Presenting you MEDUTAINMENT. Experience the joy of learning medicine. Which disease do you think is the second leading cause of death in the more developed countries like US? This disease and its agonizing reactions have been well imprinted in our minds via the film industry. Yes, you have guessed it right. It's cancer, more accurately known as neoplasia, which means new growth. So today we'll be looking at the molecular basis of a cancer. That is, we're going to understand cancer at the level of the genes. Back then, in the olden days, scientists described the functional consequences of alterations of one gene at a time. But it was so hectic to go gene by gene. So a more tractable and conceptually satisfying way to think about the biology of cancer is to consider the common phenotypic properties that are imparted to cancer cells. And that's how 8 fundamental changes in cell physiology were recognized, and they got to be the hallmarks of cancer. The 8 hallmarks of cancer are depicted in this picture. The first being the self-sufficiency in growth signals, evading growth suppressors, altered cellular metabolism, evasion of apoptosis, inducing angiogenesis, activating invasion and metastasis, enabling replicative immortality, and avoiding immune destruction. So today we'll be talking about the first hallmark of cancer, self-sufficiency in growth signal. Let's see some basic physiology first. According to normal physiology, there is always a signal. Here the signal is being produced by the growth factors, and then the signal is getting received by the receptors on the cell membrane. Next, the signal transduction takes place via several pathways, and all these signals they reach the nucleus, and there a transcription factor is formed and it gets attached to the DNA. Next, the DNA transcription takes place. Following it, translation occurs and the proteins for cell proliferation are being produced, and finally cell proliferation takes place via mitosis. And now let's check this out with a bit of animation. So the growth factor, it goes and hits the receptors on the cell membrane. Once this happens, the receptors they get activated and they pass signals via several signal transduction pathways to the nucleus. And here in the nucleus, the transcription factor is being formed. This transcription factors, it attaches to the DNA, and DNA transcription takes place following translation. Through translation, several proteins required for cell proliferation is being produced, and finally cell proliferation takes place. Any aberration in the signal pathways leads to increased or unlimited cell proliferation, leading to various tumors. As mentioned before, there are various signal transduction pathways in our body. The list of the various signal pathways is given here. Today we'll be seeing more in detail about the receptor tyrosine kinase pathway. So here we have the receptor tyrosine kinase pathway. Initially, the growth factor binds to the receptor. This leads to dimerization of the receptor. Once the dimerization takes place, the tyrosine residues present in the receptor gets phosphorylated, and the adapter protein, the RAS, gets attached to the receptor. Initially, the RAS is inactivated because of the presence of GDP. Now, the GDP gets phosphorylated to GTP in order to activate the adapter protein. Now, the activated RAS, it activates 2 pathways, the P13K pathway and the RAF pathways. And then the transcription factors, they get formed and cell cycle progresses. Once the process is over, the activated RAS turns back into inactivated RAS, since the GTP hydrolyzes to GDP, and the cascade P13K and the RAF pathways comes to a halt and cell proliferation stops. So now let's see the various mutative mechanisms in the receptor tyrosine kinase pathway via which the tumor can hit the body. So the mutation can be present at different levels. It may be at the level of the receptor, or at the level of the adapter molecule RAS, or at the level of the signal transduction pathways, the P13K and the RAF pathways, or at the level of the transcription factors, or maybe at the level of the cell cycle progression via cyclins. So now let's see mutation at the level of the receptor. So in lung adenocarcinomas, there is a point mutation in ERBB1 gene. So this ERBB1 gene in general, it codes for the EGF receptor. Now when there is a point mutation, it causes the production of a constitutively, that is all-time active EGF receptor. This all-time active EGF receptor, even without the presence of the growth factor, it remains active and it sends the signals to the nucleus for the production of transcription factors and the cell cycle progresses. Similarly, in breast carcinomas, there is an amplification in the ERBB2 gene. ERBB2 gene in general codes for the HER2 receptors. So now when there is an amplification, it leads to overexpression of the HER2 receptors, and all these receptors, they receive signals from the growth factors and they keep giving signals to the nucleus for the production of more cells. Similarly, in EML4 and ALK fusion, the gene rearrangements like the deletion on the chromosome 5, it leads to fusion of ALK with EML4 gene. The EML4 ALK protein has a constitutive tyrosine kinase. So why do we have to know the oncogenes and the oncoproteins? Yes, for creating a targeted therapy. For example, in breast carcinoma, the treatment can be targeted to those particular receptors like the HER2 receptors. That is, antibodies or drugs can be created to block the HER2 activity, thereby leading to the cessation of the tumor growth. Similarly, in lung adenocarcinomas, the targeted therapy against ERBB1 and EML4 ALK fusion gene mutations have been discovered. Unfortunately, none of these targeted therapies cured advanced lung cancers. This is because the treated patients have found to have acquired activating mutations in some other signaling molecule. So this sidesteps the effect of the drug. So the patient becomes resistant to the targeted therapy against these ERBB1 and EML4 ALK mutations.